Hello everyone, it's Mimi here. BMS working in the NHS. And today's video will be about tissue processing and embedding, which is part of the IBMS specialist portfolio in cellular pathology. So, just a quick introduction to this video. It will be short and sweet, I promise. I will discuss rationale and aims, learning outcomes, indicative content, resources, submission of evidence, Q&A suggestions, some evidence ideas, reflective statement, and some helpful tips to make your portfolio better. With all that said, I hope you will enjoy the content of my video and I hope you will learn a lot. So let's dive into this submodule. Rationale and aims of this submodule outline the general information and expected knowledge and competence that needs to be achieved when this submodule is completed. These are relevant to each submodule. For the tissue processing and embedding submodule, the expectations are that the candidate will have knowledge and competence to process, embed, quality control of routine histological tissue samples. Will gain clear understanding of the principles and practice of tissue processing and embedding. Will understand the role of processing and embedding in the investigation of key pathological conditions and how these may be investigated histologically. The learning outcomes demonstrate candidates' knowledge, skills, and competence in this submodule. They also include demonstration of the understanding of scientific principles, laboratory procedures, quality assurance, and professional conduct. They allow candidate to work autonomously and solve problems independently. Let's explore the first learning outcome, which is for the candidate to explain the principles and practice of paraffin wax tissue processing for a range of tissues. What are the principles and practice of wax processing? What steps are involved during tissue processing? What reagents are used during processing and why? Consider each processing schedule, reagents used during processing, reasons why those reagents are used, quality of reagents used. Consider factors affecting processing like vacuum, temperature, or agitation. Also consider different processing schedules like rapid schedule, biopsy schedule, fatty or routine tissue schedule, and so on. The second learning outcome is for the candidate to discuss the influence of processing schedules on the quality of sample preservation for subsequent analysis and give an example from practice. How can processing influence subsequent tissue analysis? Is every tissue sample processed the same way, or are there different processing schedules? What would happen if wrong tissue schedule was selected during tissue processing? Consider types of processing schedules, the importance of selecting the correct processing schedule, reasons why there are different processing runs, tissue size and density, effects of incorrectly processed tissue on subsequent analysis like IHC, molecular testing, routine staining. The third learning outcome is for the candidate to discuss how to troubleshoot processing errors. What errors could occur during tissue processing? How could those be resolved or prevented? What effects could they have on subsequent analysis and tissue reporting? Consider the key areas of processing, different types of problems, possible measures to resolve problems, but also steps taken during troubleshooting. Try to demonstrate the application of your knowledge and skills in this area into practice to resolve problems. The fourth learning outcome is for the candidate to compare the advantages and disadvantages of different processing reagents and schedules. What reagents are used during processing? What are their alternatives? What are their advantages and disadvantages? Consider each reagent used during processing, alternative reagent that could be used, their advantages and disadvantages. What are the advantages and disadvantages of different processing schedules? Consider turnaround time, cost, quality, artifacts, health and safety considerations, reliability, consistency, flexibility, throughput, and so on. The fifth learning outcome is for the candidate to discuss the effects of heat, pressure, and vacuum on tissue processing times and tissue morphology. What effects does heat, pressure, vacuum have on tissue processing times and tissue morphology? Consider how heat, pressure, and vacuum speed up tissue processing, effects on tissue morphology like shrinkage, hardening, brittleness, distortion, antigen damage, uniformity, speed of infiltration or penetration, quality of staining, and so on. The sixth learning outcome is for the candidate to explain the principles and practice of resin processing. What are the principles and practice of resin processing? What steps are involved during resin processing? What reagents are used during resin processing? Which samples can be resin processed and why? Consider principles like steps and reagents used during resin processing, the practice like superior morphology, antigen preservation, ultra-thin sectioning and so on. Also consider the advantages, disadvantages, safety considerations, and quality of resin processing. The seventh learning outcome is for the candidate to explain the principles and practice of a range of tissue embedding techniques, and discuss potential errors, and their clinical impact. What are the principles and practice of embedding? Consider embedding techniques like paraffin wax, cryo-embedding, and resin or plastic embedding. What could be the potential errors associated with embedding? Consider orientation, air bubbles, sample mix-up, poor tissue support, presence of staples or sticks, tissue not flat and so on. What could be the clinical impact of poor embedding? Consider tissue loss, misdiagnosis, delayed diagnosis and treatment, unnecessary procedures, compromised staining quality, distress, turnaround times, effect on subsequent analysis and so on. The eighth learning outcome is for the candidate to appraise the correct orientation for embedding a range of tissues, demonstrate candidate's application of this in practice. How would you orientate tissue during embedding? What tissue types require specific orientation during embedding? Consider skin, hollow organs, tubular structures, inked or bisected samples, samples with specific special instructions and their reasons like muscle, nerve, alopecia, or temporal artery samples. The ninth learning outcome is for the candidate to discuss the risks and hazards associated with the use and disposal of processing reagents and embedding media. What are the health and safety risks and hazards associated with reagents used during processing and embedding? Are there any safer alternatives that could be used? How would you minimize health and risks and hazards? How would you dispose processing and embedding reagents and why? Consider health hazards, safety hazards, environmental hazards, COSH, SOPs, PPE, risk assessments, training, regulatory bodies, waste segregation. How would you mitigate those risks and hazards? You're using a table. Those are the 9 learning outcomes that have to be met for this submodule. Indicative content highlights knowledge and understanding requirements that might be assessed during the Viva. They may or may not be demonstrated by the learning outcomes or candidate's evidence. In the tissue processing and embedding submodule, candidate is expected to have knowledge and understanding relevant to tissue processing and embedding for histology samples. How do you process and embed routine histology tissues? Why are those processes used? What other processes can be used? Understanding relevant to EQA schemes for tissue processing and embedding and be able to interpret and act upon the results. What are the EQA schemes for tissue processing and embedding? What do the EQA results mean? How would you interpret them? How would you address poor EQA results? Resources section on the Brightspace contains some very useful information. Submission of evidence on Brightspace informs the candidate what evidence needs to be submitted for each subsection. Candidate needs to submit answers to questions that were set by the training officer. I will share some examples of possible questions later on. Candidate also needs to select an additional piece of evidence relevant to this subsection. I will also share with you some ideas of possible evidence later on in this video. Each submodule requires the candidate to submit their reflective statement relevant to their practice and training in each submodule. Questions should be set by the training officer. They need to be based around the knowledge requirements, relevant for each subsection. They should allow the candidate to demonstrate their understanding in each subsection. The questions should be specific and relevant to the candidate's in-house practice. They should also allow the candidate to demonstrate their specialist knowledge. If you would like to see specific example questions, please have a look at the video for trainers. Link will be included in the video description. These are some ideas you could explore for your chosen piece of evidence. and some more ideas for your chosen piece of evidence. Each submodule also contains a reflective statement which is asking you, the candidate, to summarize your role in the laboratory in the context with each submodule. How do you demonstrate that you can draw a conclusion from what you have learned, reflect on your current practice and how it can change in the future due to gained knowledge? Think about what is your laboratory in context this submodule. What impacts can your actions have on your colleagues, department as a whole and for the patients? You also need to write a personal reflection on your training and why you chose your evidence. This is very personal to your own circumstance. It should demonstrate your ability to be self-aware when analyzing the gaps in knowledge. You can use it as an opportunity to reflect on aspects of your training, application of your gained knowledge and skills, and how you have achieved your goals. Think about what your evidence demonstrates. Why did you choose this specific evidence for this submodule? Also write what your training was like when it comes to this submodule and how your practice changed due to your training. The reflective statements can also be used as a CPD evidence in case you are chosen to be audited by the HCPC. I would also like to share with you some helpful tips that can make your portfolio look really good. Try to present the information in various formats such as tables, pictures, photographs, diagrams, flowcharts, graphs or even videos, if you can. This nicely breaks down a lot of text that is usually present in specialist portfolios. Try to make it a little bit more interesting to the reader, which will be your training officer and eventually the assessor. Try to put yourself in their shoes. How would you like to read hundreds of pages of plain text? There is a saying I really like, and it goes like this: a picture is worth 1,000 words. I will make a video. I will share with you my evidence I used for my portfolio back in the day. Hopefully that will help. Also try to use various types of evidence, not just one type, like case studies, for each submodule. You could use experiments, case studies, annotated results, annotated photographs of you performing tasks, evidence of troubleshooting, annotated EQA results, nonconformances, validation and verification evidence, comparisons of different approaches and their pros and cons, control material production, certificates of course attendance with reflective statements, annotated images or charts, and so on. Try to bring in your personality. Think about the ways you like to learn best. If you like experimenting, perform experiment. If you like troubleshooting, perform troubleshooting. Choose something that demonstrates your understanding, knowledge, skills, and how you apply this in your practice. And one more thing, keep the learning outcomes in mind when you selecting your evidence. Good evidence will tick off several learning outcomes. And this brings me to the end of today's video. Hopefully you now have a better understanding about the learning outcomes for this submodule. And I hope you also have a better idea what evidence you can choose to demonstrate your competence. And my final take-home message? Keep those learning outcomes in mind when answering questions and selecting your evidence. Bye for now.